Hi, I'm Shane Warnicki. I'm a detective here at the Anoka County Sheriff's Office, and I work in the crime scene unit. Here at the Anoka County Sheriff's Office this week, we're celebrating Forensic Science Week. So I wanted to take some time and show you some of the stuff that we use here in the crime scene unit. Our crime scene unit consists of 6 detectives. We work county-wide, and we go to basically any crime scene that we're requested to. Some of our main calls are burglaries, shootings, death investigations, all kinds of stuff. So why don't you come take a look and I'll show you some of our equipment. Here we are at my crime scene unit, Anoka County Sheriff's Office squad car. This is a squad car that's a little different than what people normally see out on the road. We don't have the cages in the back. We're typically not transporting people. We're typically not even arresting people. Our squad cars are full of equipment that we use on various scenes. We have all sorts of different tools and toolboxes, which we turn them into kits. This one is actually a fingerprint kit, which I can show you a little bit more of in a few minutes. But we have a kit for basically everything, all of the disciplines of investigating crime scenes. So that's how our squad cars are different than most squad cars that people are used to seeing on the street. Do you guys remember trying to stay awake in high school math class? Well, I didn't stay awake. That's why I had to relearn all of my math so that I could determine some shooting reconstruction and impact angles of bullets. It's one of the many disciplines that we do here in the crime scene unit. No, I'm not a doctor. I'm wearing this to protect this item here, this bottle, from my own DNA. The science in detecting DNA has become super sensitive that even breathing on an object, I can inadvertently put my own DNA on it. So in crime scene investigation, we are continually gathering DNA from various surfaces and different sources on particular scenes that we go on. The way we do that is we open 2 swabs in there. This here is sterile water. I would open that, and it's as simple as placing some sterile water onto the cotton swabs and then swabbing the surface of the item. On a bottle like this, we want to swab where someone would be likely to touch it, like kind of around the sides here. Did you know that every human being on Earth has a unique fingerprint? I'm sure you'll learn more about that here on Forensic Science Week at the Anoka County Sheriff's Office. Fingerprinting is just one of many tools that we use to potentially identify a suspect in a crime scene. We'll show you more about that a little later. Yep. Just as I suspected, bodily fluids. This here is an alternate light source. We use it to search for bodily fluids on various surfaces for a variety of reasons. We use it with different colored goggles, and it reveals different bodily fluids. So without the light on, you see nothing here. But once I turn the light on and shine it on it, now you're seeing what I saw. Stains are luminescing in various places on this bed sheet here. Blood! Blood is everywhere. Here's my best guy, Chris, to tell you a little bit about blood spatter analysis.
Hi, my name is Detective Chris Fahey. I'm with the Anoka County Sheriff's Office Crime Scene Unit. I'm here to talk to you about blood. One of the disciplines that we have is blood stain pattern analysis. This room right here is set up for detectives to practice because this is one of the disciplines that if you don't practice, you kind of lose those skills. So what we do is we put up Tyvek, we put up cardboard, craft paper, and then we throw blood around for us to practice because when we get on a real scene, you want to have your skills honed the best you can. If you look around here, you see on the floor we have a drip trail, which is blood that's dropped as the suspect or an object is dripping blood off of it. On the wall, we have a spatter stain. And then we have here what we call a swipe, or it could be a wipe, which is something moving through blood. We can set up a variety of trainings in here. Like I said before, when we get to a scene, you want to have all this stuff dialed in, and this is the way we practice. As you can see here, Detective Nick Smith of the Anoka County Sheriff's Office Crime Scene Unit is Photodocumented Vehicle. This vehicle was used in an armed robbery, was stopped by the police, the driver was taken into custody. Now the Crime Scene Unit has been called to go ahead and photodocument and process the vehicle. As explained earlier, part of our processing efforts include fingerprinting a vehicle or any other articles that might be of evidentiary value and have fingerprints on them. Detective Smith is using a white fingerprint powder because it contrasts against the darker colors of the vehicle. So as he applies the powder, any latent fingerprints that are on the surface will show up. Detective Smith is now applying fingerprint tape, and he's going to lift the fingerprints. So as he's applying the tape there, he presses down. Now he's labeling it. And when he lifts these, he'll apply them to a clear plastic slide, which will be submitted to the lab. As you can see, he's applying the clear plastic tape to the clear plastic slide. The key to this is not to have any bubbles or wrinkles in it. After all exterior processing is done in the vehicle, we move to the interior of the vehicle. Detective Smith is collecting a touch DNA swab from the steering wheel, which will be sent to the lab for later analysis. We typically do all exterior processing first, which will be DNA and fingerprinting. At this point, Detective Smith is beginning his evidence collection. The first thing he is collecting inside this vehicle is the handgun, which is reported to be used in the armed robbery. Every item in this vehicle has been photo-documented prior to evidence collection. He's removing the weapon, putting it in a gun box, where he'll photo-document more in great detail. So other items of interest for us in this vehicle would be the Gatorade bottle in the center console. That can be swabbed for DNA. You also notice there's a glass pipe, typically associated with drug use in there. That can also be swabbed for DNA, or it could be fingerprinted. Before handling each item of evidence, he changes the exam gloves, and each item is put into a new evidence bag. Here we're using paper bags because it's more conducive to collecting DNA and fingerprinting.
Welcome to the Midwest Regional Forensic Laboratory. I'm Lab Director Scott Ford. This week we're celebrating the 7th Annual National Forensic Science Week. In these videos, we're going to be showing you how evidence that's collected from crime scenes is processed in our nationally accredited forensic laboratory. The Midwest Regional Forensic Laboratory is a partnership between Anoka, Sherburne, and Wright counties, and we're housed inside of the Anoka County Sheriff's Office. At the Midwest Regional Forensic Laboratory, we have 3 main areas of testing: biology, drug chemistry, and latent prints. And during this week, we're going to have videos that will have demonstrations of how evidence is handled and processed in all of those different areas of processing. So welcome to the Midwest Regional Forensic Laboratory.
As authorities work hard to get illegal drugs off the streets in our communities, the Drug Chemistry section of the Midwest Regional Forensic Laboratory has seen an increase in requests for service from 2018 to 2019. Even with the increase of cases, the scientists at the lab have been able to produce a quick testing turnaround time of approximately 10 to 14 days. We will highlight a few of the tests that the drug chemistry scientists perform on a daily basis in order to assist detectives in building accurate cases. There is a large variety in the types and sizes of drugs brought into the lab for testing. At times, it is necessary to break substances into smaller fragments so that the proper tests can be administered for identification. Once the substance is at its optimal size, a preliminary color test is done by mixing a sample of the substance with different reagents in order to determine the reactions. Based on the colors that appear after the mixing, scientists can begin to suspect what type of substance it is. Another preliminary test is an ultraviolet visual spectrometer. UV light will pass through the substance and measure how it absorbs light at different wavelengths. Data from this test can then be compared to data from confirmed substances to obtain a preliminary result. One of the identification confirmatory tests that can be done is on the Fourier Transform Infrared Spectrometer. This device will shoot a laser through the substance and then measure the amount of light absorbed by the sample. Using predetermined algorithms, this machine will use the data to present a picture of information which again can be compared to known samples of drugs. A second identification confirmatory test is the gas chromatograph mass spectrometer. This machine runs 2 tests on a sample to produce results that can be analyzed. The first part of the test is the gas chromatograph, which heats up the sample until it turns into a gas. At that point, helium pushes the gas through a column, which will break down the substance into its components. Different components will push through the column at different speeds, which is measured by the system. Once the sample has gone through the column, it then goes into the mass spectrometer, which further breaks down the components into the ions present. Data is extracted, and the scientists can compare the data from both tests in order to make an identification. While this is not a complete presentation of all of the tests offered by the drug chemistry section, we hope it provides a better understanding of the important work that these scientists do every day. Did you know that fingerprints are comprised of approximately 99% water, with the other 1% being made up of amino acids, lipids, and fats? Today we're going to see how the Midwest Regional Forensic Lab collects and processes fingerprints that help identify suspects in cases ranging from burglary to homicide. The key to fingerprint analysis is contrast. Therefore, different processes are completed in order to preserve and give contrast to fingerprints located on evidence. When analyzing a non-porous item such as a can or firearm, the first step is to use the superglue fuming chamber. This temperature and humidity controlled chamber allows for superglue to adhere to the water in the prints, which provides stability to the fingerprint. After the superglue has bonded to the item, it is then sprayed with a fluorescent solution, which will be placed under a laser for analysis. The solution will only appear under a particular wavelength of light, which can be seen with the filtering goggles. Obtaining fingerprints on porous items, such as wood and paper, requires the scientists to focus on the non-water components of a fingerprint. By soaking the evidence in a solution that shows the amino acids, lipids, and fats, scientists can then see the print on the surface of the item. After the fingerprints can be seen, they are then analyzed and compared against the prints of possible suspects or submitted into state and federal databases to determine if there is a match. Lieutenant Dan Douglas with the Anoka County Sheriff's Office will share an example of when a latent fingerprint match led to the identification of a suspect in a local homicide that shocked the community.
On the morning of November 28, 2016, an elderly male was found deceased in his home in the city of Anoka. He was found there by his adult daughter. When she found him, she discovered that the home had been ransacked and appeared to have been burgled. The victim was later identified as 95-year-old Albert Lowleen. Mr. Lowleen had suffered severe trauma to his body and face and was found deceased in his bedroom. It was of great concern to us in law enforcement, both the Anoka Police Department and the Anoka County Sheriff's Office, is the brazenness of this burglary. Mr. Lowleen lived alone. He was widowed. His wife had died about 10 years prior. Certainly Mr. Lowleen was not in what we would consider a high-risk category as to being the victim of violence or crime. Also, we were unsure as to when this could have occurred. This was at the end of a long Thanksgiving Day weekend. No one had seen or heard from Mr. Lowleen for a couple of days. There were no suspects at the time of this other than we quickly surmised that this was a burglary likely gone bad. Because this was likely a burglary, we anticipated hopefully finding some sort of evidence, whether that was through DNA or fingerprints. Because Mr. Loling had lived there alone for a long time and he was legally blind, the home was somewhat dusty. This allowed our crime scene detectives to look around and discover which areas within the home had been disturbed, which objects were likely moved from one location to the next. We also were able to find other items that were broken and misplaced. A couple of articles within the home were found to have been moved from one area of the home to the other. As our crime scene unit began looking at those items, they discovered there was likely a great chance of finding some fingerprints on those items, one item being a small wooden box. It was on November 30th, 2 days after the discovery of Mr. Loling's body, that the latent print found in Mr. Loling's home was matched to Isaiah Thomas. A review of law enforcement records found that Isaiah Thomas at one point had resided approximately just a half a mile from the crime scene. Mr. Thomas also had an extensive criminal history for burglary. The Anoka County Sheriff's Office requested the help of the Anoka Violent Crimes and Narcotics Task Force to begin surveillance on what we believe to be Mr. Thomas's home in Anoka, just off East River Road. Shortly after surveillance began, a man matching Mr. Thomas's description got into a vehicle, and that vehicle was then stopped on Coon Rapids Boulevard in the city of Coon Rapids. The suspect was positively identified as Isaiah Thomas, and he was taken into custody at that time.
Biological evidence can be a crucial piece of data to solve severe crimes such as sexual assault and homicide. The biology section of the Midwest Regional Forensic Lab is able to test samples of evidence that are less than half of the size of this blood sample. The first step in this process is for the serology scientists to test evidence for pertinent DNA samples. Once these samples have been identified, they are given to a DNA analyst for testing. Multiple processes with various reagents are done in order to release the DNA from the sample, separate cell materials from the DNA, and purify the DNA. The purified sample then goes through another test to determine if any DNA has been able to be extracted, and if so, how much. The results from that test are then used by the scientist to input the necessary amount of the sample into a system which targets 24 regions of DNA. It then dilates them. Too much or too little amount of the sample will not provide a good profile for interpretation. Finally, more reagents are added to the sample and then put into a machine which will provide the profiling results that then can be analyzed by the scientist. Once the scientist is able to analyze the results, they can then compare it against a sample from a suspect or consider entering it into the Combined DNA Index System, or CODIS. If the profile meets specific criteria to be entered, it can be ran against DNA collected within the lab or be ran against DNA in the state and federal databases to see if there is a match. Lieutenant Douglas will provide a conclusion to the Albert Loling homicide case, where DNA was also used to confirm the suspect.
An additional key piece of evidence in this case involved DNA evidence. A number of items were collected by the crime scene unit from Mr. Loling's home and later processed at the Midwest Regional Forensic Laboratory. One item, the broken end of a flashlight, was found to contain both Mr. Loling's and Isaiah Thomas's DNA. Because of the Midwest Regional Forensic Laboratory being co-located within the Anoka County Sheriff's Office, turnaround time on this case was extraordinary. Where normally we could have been waiting days, weeks, or even months, we were able to make an arrest within just over 2 days of discovering this horrible crime. Due to the overwhelming amount of forensic evidence in this case, Mr. Thomas ultimately pled guilty and was sentenced to life in prison, providing some closure and peace for a mourning family and a community shocked by this outrageous act.